So we're going to start out just with these two topics. So what are AOPs and what are they good for? At least, what do we think they're good for? So I'd like to start out with something that most people are familiar with. So back in 1962, Rachel Carson published a Silent Spring, which basically made the association between exposure to DDT and its metabolites like para-DDE and declines in sensitive bird populations as a result of eggshell thinning. And this remains today as perhaps the most well-known incident of wildlife ecotoxicology and really helped spark the environmental movement, as well as, in many respects, the mission of the EPA, at least as it relates to chemical contaminants in the environment. And so, since that time, really a central challenge for regulatory toxicology has been this question of how do we identify other chemicals out in the environment that may cause similar adverse effects, or adverse effects on humans, other wildlife species. And importantly, how do we actually do that before we see impacts on humans or wildlife populations? We don't want to have to wait until bird populations have declined to the point of near extinction before we actually take action on these things. And so the traditional approach, of course, has been whole animal toxicity testing. I mean, that's what we've done within the society for decades. That's been sort of the regulatory approach. If we're concerned about eggshell thinning in birds, we develop a whole animal toxicity test that measures eggshell thinning in birds. That's one of the endpoints in the avian reproduction study, the OECD 206 test guideline. And that's effective, works well. You know, we can screen chemicals for their ability to produce this effect. The problem with this approach is it's incredibly costly, about $250,000 to actually conduct a test. It takes over 30 weeks to perform. If we have tens of thousands of chemicals in commerce, we can't afford to screen all chemicals through this type of assay. And so, you know, the scope of application for that is somewhat limited. But if we understand how chemicals are causing adverse effects and the biological activities that lead to or are associated with progression toward adverse outcomes, this creates opportunities to use new types of data for both hazard identification and risk-based decision making. So over the subsequent decades since Rachel Carson initially published Silent Spring, there's been a lot of research done on how it is that DDT or para-DDE actually cause eggshell thinning. And while there's still some debate in the scientific community about exactly how that's occurring, one of the leading hypotheses right now is that para-DDE actually causes inhibition of cyclooxygenase, which is an enzyme involved in the synthesis of prostaglandins. That leads to decreased transport of calcium and bicarbonate into the eggshell gland lumen. That reduction in calcium flux into the lumen results in thinner eggshells, which then, of course, makes those eggs vulnerable to physical damage, breakage, crushed eggs, and as a result, you see declines in these bird populations. So if we know something about how it is that compounds like para-DDE can cause this effect, we can use new approaches to screen chemicals for their ability to produce these sorts of outcomes. And one of the ways we can do that is through the use of high-throughput toxicology tools. So now we have tools available through programs like EPA's ToxCast program, where we can screen chemicals in a large battery of pathway-based assays. So hundreds of different assays in a matter of days, screen them for a broad spectrum of biological activities, and do that for a fraction of the cost that it would take to conduct one avian reproduction assay. And get a measure from that assay that allows us to make some inference about potential hazard of that chemical. This is really the vision that was laid out by the National Research Council back in 2007 when they published their report on toxicity testing in the 21st century, and really advocated that we transform toxicity testing from a system based on whole animal testing to one founded primarily based on in vitro methods that are administered as suites of predictive high-throughput assays that assess critical mechanistic endpoints that are involved in the induction of overt toxic effects rather than measuring the effects themselves. So that was the vision laid out. A number of research programs, testing programs have responded to that vision. EPA's got their ToxCast program, which again is a research effort exploring how we use these types of high-throughput in vitro tools for risk assessment regulation. To date, this is actually a little out of date. To date, the ToxCast program has screened somewhere on the order of 3,000 chemicals now against well over 600 different pathway-based assays. Those data are all publicly available. You can go right now and type into a Google search, ICSS dashboard, have full access to those data, search by chemical, search by assay type. A lot of good information there. You've got also programs like Tox21, which is a consortium between EPA, FDA, and IEHS, NCGC, maybe I'm forgetting one or two. But basically, same sort of principle, smaller number of assays. They're not looking at hundreds of assays, but somewhere on the order of 25 assays per year, but screening 10,000 chemicals against those assays, full dose response characterization in these pathway-based assays. So I think we can reasonably argue that 21st century toxicity testing is already here. We have the ability to conduct these pathway-based assays. We have the robotics, we have the computational capabilities to generate the data, to process those data. So we can fairly rapidly and cost-effectively generate this kind of data for thousands of chemicals against hundreds of different pathways. And it's conceivable that majority of chemicals in commerce could be tested in this manner in a relatively short period of time. Now there are some exceptions. Volatile chemicals, for example, aren't very easily tested in these methods. So there's certainly some aspects of chemical space that won't be covered. But the vast majority of chemicals in commerce, we could have this kind of information very soon. So then the question is, well, what do we do with it? If I take the results from a ToxCast screening of a chemical and take them to a regulatory program office within EPA, and I say, here, look, we've got this compound. It's got very high potency to interact with cyclooxygenase. Well, they say, so what? They don't regulate enzyme activities. They don't regulate receptor binding. They don't regulate binding to DNA. They regulate effects on human health, effects on survival, growth, reproduction in wildlife populations. So how do we translate from these changes in pathway-based bioactivities to something that actually matters from a regulatory perspective, from a societal perspective in terms of protection goals? So that's really what the adverse outcome pathway framework is about. It's making that translation. So an AOP is simply a conceptual framework that portrays our existing knowledge concerning a linkage between some molecular initiating event where a chemical actually interacts with some molecule in an organism to cause a perturbation in that organism's biology, how that perturbation then cascades across levels of biological organization, causing changes in the biology that ultimately lead to an adverse outcome that occurs at a level of organization that we actually care about from a risk assessment or regulatory perspective. So an AOP essentially helps us organize what we know and utilize that knowledge to support the use of some of these alternative mechanistic data sources in risk-based decision making. So if we take this example, and now we've got this data set, we show that this chemical is very potent to interact with cyclooxygenase. If that regulator has access to this AOP knowledge that says cyclooxygenase inhibitors actually have potential to lead to eggshell thinning in birds, for example, now the regulator can say, aha, I've got some idea of what the potential hazards are associated with this compound. This might be a compound for which it would be worth investing in doing an avian reproduction study. These are the kinds of hazards I might expect. If we have access to, again, things like the ToxCast data set, now if we're concerned or we want to think about, we want to order some avian reproduction assays, which chemicals should we focus on? We can go through, rank that list of thousands of chemicals based on their potency against this particular target, decide which of those should be priorities where avian reproduction outcomes may actually drive the risk assessment. So it's a way to prioritize what kind of testing we might do, identify a set of chemicals for which there might be reason to expect these kinds of outcomes. We can use these different key events that are identified along this pathway to help suggest some alternative tests. So say we've got this list of 1,000 chemicals and we've got 50 of those that are active in this assay. Well, that's in vitro. We don't know if they're rapidly metabolized. We don't know how well they're taken up by the organism. Do we want to conduct a 30-day or a 30-week avian reproduction assay to figure that out? Not necessarily. But based on these key events, could we design a shorter in vivo test? Maybe after a couple days, look at prostaglandin concentrations in the eggshell gland mucosa, get an idea of whether that chemical is actually getting into the organism, causing the predicted sorts of effects that could ultimately lead to that outcome. These key events also suggest biomarkers that we could take a look at in wild populations of birds, or birds that we cage out in the environment, or fish we would cage out in the environment, for example. That might give us some early warning or diagnostic potential to say we've got contaminants out in the environment that are causing effects that we think could be associated with adverse outcomes. That overall pattern of effects gives us some diagnostic potential to say if we see this kind of profile or motif of biological effects, might point us in the direction of chemicals that inhibit cyclooxygenase, give us a chemical space that we might want to look at in terms of monitoring. As we break adverse outcome pathways up into this series of key events, it also helps us to systematically evaluate the taxonomic relevance of these different pathway-based activities, which taxa this actually matters to. So if we take the case of cyclooxygenase inhibition, is that enzyme even found in invertebrates? Is it found in plants? Is that target there for the chemical to interact with? That can give us some insights into what the potential relevance domain is for potential effects of that chemical. If we look at these key events at higher levels of organization, think about the physiology of the organisms. In the case of eggshell thinning, what species are actually laying hard-shelled eggs? That, again, gives us some ability to frame the domain of applicability of that particular AOP. Again, high-throughput screening, although it's maybe one of the main drivers behind the interest in AOPs right now, it's certainly not the only relevant source of mechanistic data. The toxicology research community has been studying mechanisms that underlie toxicity for decades. There's lots of this information available in the literature, and much of that is currently underutilized for regulatory applications for, again, the same reason. How do we translate those mechanistic data and those mechanistic understanding into a prediction of hazard and outcome that we actually care about? So the more we can make that linkage, the better we can utilize these alternative types of data that already exist. We can also think about applications of these adverse outcome pathways in the context of environmental monitoring. So we've been developing a number of approaches in our lab where we can take something like a water sample, it could be a sediment sample, it could be really any environmental media. One approach is to do some chemical analysis on that. Through the use of various pathway-based tools, we can make connection between chemicals and specific pathways that are perturbed. So a lot of this information is curated, for example, in the comparative toxicogenomics database, or you could compare this directly against the ToxCast database. Say, I've got this chemical, it interacts with this pathway. AOPs then help us translate that pathway interaction to a prediction of hazard, to identification of endpoints that we could use for targeted monitoring. Likewise, we could take an ambient water sample, sediment sample, so on, extract that run that directly in these high throughput pathway-based assays, screen for bioactivity, once again, use the AOPs to make the link to hazard endpoints that we might want to use for monitoring purposes. So overall, in terms of 21st century regulatory decision making, I would argue the data are there. We have these emerging sources of high throughput screening data. We've got entire libraries of scientific publications, decades of mechanistic toxicology research that we can tap into. So really the scientific challenge of the 21st century is how do we make effective use of those data and our wealth of existing scientific knowledge to support regulatory decision making. And we think AOPs are part of that solution. They're not the only solution. There are other things we need to bring into play, but that's why we're interested, and that's why we're really here, and hopefully that's why you're here as well. So in general, this adverse outcome pathway concept is not really new. It's been around in the ecotoxicology community since at least the late 80s. I think you could go back to abstracts from the CTAC meeting. You probably wouldn't find the term adverse outcome pathway, but you would certainly find descriptions of research and approaches that look a lot like what we're talking about today. There was a Pelston workshop back in 1992 looking at how we make better use of biomarkers in ecotoxicology. Really talked about the need to make these linkages across levels of biological organization, link this to the things that actually matter for regulatory decision making. Throughout the mid-90s, there was a lot of interest in making these connections as a way to make more effective use of quantitative structure-activity relationships, predictive toxicology. You can look in 2004, there was publication talking about this concept as a way to make more effective use of in silico and in vitro data in toxicology, and at the time sort of termed that toxicity pathway. With the publication of the NRC report back in 2007, they really took those same concepts, although kind of narrowed the definition of toxicity pathway a little bit and kind of focused on these cellular perturbations. And so that kind of led to adapting this term adverse outcome pathway to kind of, again, capture that original vision of going across these levels of biological organization. But this concept's been around for a very long time, and I think what's really changed is, again, this shift in thinking and paradigm where we want to move to using those mechanistic data as a basis for regulatory assessment. If we're going to do that, we have to be able to translate that to the currency that's meaningful from a management perspective. And so that's really driven the interest in this concept. So until recently, the AOP development has really been an ad hoc process. It's something that researchers would sort of have in the back of their head, or maybe we'd sketch it out on a napkin or make a PowerPoint slide. But if you take that to a regulator and say, yeah, cyclooxygenase activity really matters, when can they actually trust that there's actually a scientific foundation behind that? And I'm not just feeding them a line of BS. How do they understand what is the appropriate biological and toxicological domain of applicability of those relationships? What taxa does this relationship apply to? Does it apply to all sexes, all life stages? How can they understand how best to apply that? And how can we present all that in a very systematic and transparent manner? And that's this question of how do we describe these and present these in a systematic and transparent manner is really how the AOP framework has evolved over the last several years. It's been the real driver. Concept is largely the same, but the formalization of that concept into a process that we can use and share among the community has really changed. One of the key evolutions in that process of formalizing the AOP framework has really been interactions with the human health toxicology community. They've also been using basically the same concept for a long period of time now, largely using it in risk assessment as a basis for evaluating species concordance. So can animal data be reasonably used to predict human health outcomes? And so much of what they were trying to focus on is laying out these different key events in a chemical's toxicity and then evaluating whether those key events that they see in an animal model like a rat actually apply to a human. To say, are those data actually useful for human health risk assessment? Those assessments were done in a very chemical-specific manner. So it's a chemical by chemical sort of assessment. And because these were for risk assessment and regulatory purposes, there was a strong focus on very systematic documentation of weight of evidence and regulatory application. And so they had very formal incorporation of Bradford Hill considerations, which we'll go into, as a way to provide weight of evidence, confidence in the decisions that they're making based on this. And so as the international community started to form around how we're going to describe adverse outcome pathways, they really brought in and incorporated many of these concepts of weight of evidence assembly and weight of evidence evaluation into how we go about describing adverse outcome pathways. So in terms of an international effort toward formalizing the AOP framework, that really began around 2012 with the launch of OECD's Adverse Outcome Pathway Development Program. In 2013, OECD published the first guidance document on how to actually formulate an AOP description that gave conventions and terminology, defined the information content that we want to put into an AOP description, described the process of weight of evidence evaluation, started to introduce some of the guiding questions, and really tried to introduce standardization and rigor into how we actually go about describing these different pathways. Since the publication of that original guidance document, over the course of the next year or so, our group, others within the scientific community, took and applied those guidance, put it to practice, and found some things that we didn't like, some things that we wanted to change. And so we revised that guidance. That led to the development of the Adverse Outcome Pathway knowledge base. We'll talk about some of the reasons for that in a bit. And we developed what's called the User's Handbook Supplement to the OECD Guidance, which is really the evolution of those guidance based on those initial case studies, based on moving that framework forward. So later today, you're going to hear me talk about key event relationships. Those don't exist in the original guidance document. So things have changed quite a bit. So if you go to look for guidance, you might want to refer to the 2013 guidance as an initial historical document, but this is sort of the current state of the art as far as the AOP framework. And so that user's handbook and the wiki are really informed by a number of key principles, and these really guide AOP development as it exists now. One of the first principles is that adverse outcome pathways are not chemical specific. This sometimes takes a while for people to kind of wrap their heads around. We're not trying to describe what any single chemical does. What we're trying to do is generalize across chemicals and say any chemical that perturbs a given molecular initiating event with sufficient potency and duration can cause these types of effects. So an AOP is essentially a biological motif of failure. It's a reproducible pattern of failure that we expect to see if you perturb this target with enough potency. As an example, here we have an AOP for acetylcholinesterase inhibition leading to mortality. We're not trying to describe a separate AOP for every organophosphate pesticide and every carbamate pesticide. We don't need that. We can start here at acetylcholinesterase inhibition, describe that downstream biology. So in describing an AOP, we don't need chemical-specific information. Caveat here is that as soon as we want to apply that AOP in a chemical-specific predictive risk assessment, we have to take chemical-specific properties into account. Those are going to dictate how much of that chemical can get to the organism, where it's going to distribute in the organism, whether it's going to be metabolized or not. All that will affect whether we have enough severity or duration and potency severity at that molecular initiating event to really drive that pathway. Second key principle of AOP development is that AOPs are modular. We describe every AOP using two basic building blocks. The first of those are what we call key events. These are basically a functional unit of observation or verification. Each of these key events is some observable change in biological state. So by definition, every key event should be measurable. And every key event should be essential, but not necessarily sufficient. By essential, we mean it has a causal relationship in the pathway such that if you block any one of those key events from happening, you'll prevent the downstream events from happening. Now that's assuming you've just got one AOP that you're working with. Certainly that downstream adverse outcome could happen as a result of a different pathway intersecting with this. But in general, these are causally related, so if you block one of these, you prevent the downstream events from happening. Since these are basically measurements that we can make on a biological system, in describing the key events, we want to describe what biology we're actually measuring, so what is that change we're interested in, and then we want to define what methods can actually be used to make that measurement. How can we go about doing that? And then what's the taxonomic applicability, life stage, sex applicability of that particular measurement or change? When we talk about key events, there are two specialized types of key events that we talk about. These are still, again, measurements that are essential to the pathway, but they're specialized in that they sort of form the anchors of the AOP. One of those is the molecular initiating event, which is really a specialized key event that represents the initial point of chemical interaction with the biological system. By definition, because it's a chemical interaction, it always occurs at the molecular level. And this is really the point at which chemical-specific properties intersect with the biology. So that's sort of the interface with the chemical universe is at the molecular initiating event. The adverse outcome, again, is a specialized type of key event that's generally accepted as being of regulatory significance on the basis of correspondence to an established protection goal or equivalence to an endpoint that's currently accepted in a regulatory guideline toxicity test. We define it that way because we don't want to get into the decades-long debate about what actually constitutes adverse. This is a pragmatic, practical sort of definition for this. We want to convert it to something that people are comfortable making regulatory decisions on. If we get very high confidence AOPs, there may come a time when inhibition of an enzyme activity is enough to consider it adverse because we've got enough scientific understanding to say these are the outcomes we'll expect. But ultimately, we want to make that case. The second key building block that we talk about in terms of AOPs are what we call the key event relationships. These are basically the functional unit of inference or extrapolation along the AOP. So they basically define one key event as being upstream or causing some downstream key event, which is an effect of that upstream. So these key event relationships really say that we think we have enough knowledge that the state of the upstream key event provides us some ability to predict or infer what's going to happen next. So these key event relationships are basically depicted as these arrows in the standard AOP diagram. And people tend to put a lot of emphasis on the key events because they're these big boxes and they have text in them and whatever. But really it's these edges, these arrows between those key events that are really the heart of the AOP in terms of its predictive application. And so because we're using these key event relationships to infer something about this downstream event based on a measurement or prediction or estimate of that upstream key event, these are supported by biological plausibility, weight of evidence, and quantitative understanding. So that's the kind of information we want to capture in trying to describe these key event relationships. We assemble that weight of evidence using, again, considerations that were incorporated into the mode of action framework from human health. which were adapted from the Bradford Hill considerations that were used for assessing causality in epidemiology studies. But we want to take into account things like biological plausibility, the essentiality of the events. So if we block those events, can we prevent the downstream events from happening? Concordance of the dose response, the time course, frequency with which we observe the different key events. Overall consistency of the data in terms of if we look at multiple chemicals that interact with a particular target, do we see the same sorts of effects over and over? And within the applicable domain of that AOP, if we expose different species, for example, or different life stages, if they're within the applicability domain, do we see that pattern reproduced over and over? That all contributes to weight of evidence. The third principle of AOP development is that adverse outcome pathways are a pragmatic, functional unit of development and evaluation. And this gets to one of the questions that was raised earlier. We don't think that these pathways are that simple. It's not like if you interact with the estrogen receptor, there's this one chain of events and that's all it does. Estrogen receptor does all kinds of things in the body. And so we'd have all kinds of branches. If we, as a scientist, if we tried to describe every one of those branches, A, we don't know how we'd ever know when we're done, because there's always new things we're learning every day. It's an incredibly daunting task to try and describe every outcome in one species, let alone expand that to multiple species, multiple life stages, and so on. So it gets incredibly daunting to put all that information together. And so we're going to break this down into tractable units that I, as a scientist, can wrap my head around. I can assemble the evidence for those key event relationships, put that together. It's a tractable group of information that somebody can actually evaluate, make some judgment on how well we understand the confidence along that pathway. So essentially, any given AOP can be thought about as a route that gets you from point A to point B. It's not the entire map. It's not every possible route to get you from one place to the other. It's one particular path. Okay? So we recognize that for most real-world applications, AOP networks are actually going to be the functional unit of prediction, because we don't work in this very simplified sort of space. So we know that in the real world, typically we're exposed to multiple chemicals. There's lots of chemicals out there in the environment. We have lots of chemicals in our bodies. So these AOPs are not going to be triggered in isolation. Multiple chemicals may be triggering multiple AOPs. Even if we deal with single chemical exposures, again, if high throughput screening has taught us one thing, it's that chemicals have lots of different biological activities, particularly as you move through a dose space. Even very specifically acting chemicals, if you push them to a high enough dose, are going to start hitting other targets. So we have to think about more complex interactions. We can take this basic, simple unit of describing kind of these linear AOPs and start to build that more complex understanding. So here we have an example of 4 different AOPs linking different molecular initiating events to a common adverse outcome of impaired reproduction in fish. And what you see as you start to look across these pathways is that you've got certain key events. They're actually shared among all those pathways. These all start from different perturbations, but they converge on a common key event. You have common key event relationships downstream of that. And so as we build each of those pathways separately, what we really start to build is an adverse outcome pathway network, where you now see you've got 4 different molecular initiating events that feed in at different key events and ultimately converge and cause same type of adverse outcome, same pattern downstream of this sort of central key event node. So by building these modular AOPs, we gradually start to describe the greater complexity of biological interaction, capturing it in this modular network context. So this is really where AOPs start to meet systems biology. We can capture that network thinking to look across taxa. So again, we can use these AOP networks not only to capture the branches that come from different molecular initiating events, but also how those common molecular initiating events may branch off to different outcomes, again, in different taxa, in different life stages, and so on. So the framework can capture that information. The final principle of AOP development is that adverse outcome pathways are what we call living documents. So AOPs are nothing more than a way of organizing knowledge. As methods for observing biology continue to evolve, things that we couldn't measure 10 years ago, now we can measure. So that can introduce new key events that we could incorporate into an AOP. As new experiments are published, we have new weight of evidence that can be used to either support or reject those key event relationships that we built into the pathway. We could add new AOPs altogether or new branches in these AOP networks. And so because there's always new information we can add to these, there is no objective complete AOP. We could continue to do research and continue to add information to that description basically for infinity. So that's good for us research biologists, right? But because of this, really we're not concerned with when you're done. What we're concerned with is when are these useful? And we would argue that even with fairly limited information, AOPs can have use for certain applications. So if you're at this level that we'd call putative AOPs, where you've got a hypothesized series of key events that primarily supported either based on correlation or just biological plausibility, maybe you could apply that to something like hazard screening or prioritizing testing because you're going to follow up with additional tests. If you want to apply it to very detailed quantitative risk assessment, you better have an AOP that's down in this end of the scale where you've got very well-defined quantitative understanding of exactly how much perturbation gets you from one key event to the next and allows you to take some key event measurement and make a very precise prediction of the severity or the probability of your adverse outcome. But building these quantitative AOPs is not possible for every AOP because to build that takes a lot of time, a lot of data, a lot of resources. We don't necessarily have that for everything. And so all of these different stages of AOP development have potential utility. The level of development that we actually desire or require is going to depend on the type of application. We're going to get you ready to move into a hands-on exercise. So the exercise itself, we're going to give you some handouts. They're going to have some background information, kind of scientific information, background biology, along with some data, so some figures and tables and so on, kind of things that we're used to looking at. What we'd like you to do as part of the exercise is first take some time to read through the case study within your groups. Then among your group, discuss and identify what you think are key events in the pathway, so kind of sketch out an AOP, and then as a group, sketch out an AOP diagram that you'll then sort of present to the rest of us during the discussion that will follow. So before we jump into the exercises themselves, Dalma's going to come up and give you a little bit of background and address some of the frequently asked questions that relate to sort of mapping out that initial AOP and defining those key events.
So Dane did a pretty nice job, going through the AOP, and I myself participated in this training. So I think what's interesting is to sit through this training from perspective of someone who actually did not participate in development of this concept, not directly. So really I came into this group as a user. So I remember first time I took the training, Dane introduced the principles of AOPs and all of that, and it all seemed very clear to me. And then we got to the case study that we are kind of offering you today, exercise, and then suddenly all of these questions emerged. And a lot of the questions, were very specific because the introduction is not necessarily very specific. So I'm going to talk today about some of the questions that will emerge for sure, a subset that commonly emerged through the process of actually engaging in this case study that you guys are going to engage in today. And one of the first questions that you engage with is how many? Well, how many elements should I have in this AOP as I'm developing it? And there's no really good answer, but there's one answer we can give you. What is the minimum number of elements that you should have in an AOP? And the answer to that is 3, right? You need to have some kind of initiating event, and then you need to have some adverse outcome, and then you need to have key event relationship connecting the two. And this is something that's hard as a user. As you come into it, you kind of struggle with the idea that key event relationship is an element as well. That's very important to remember, even though in this exercise that you're going to do first today, you're mostly going to focus on key events and the AOP development as a whole. Later, you're going to talk more about key event relationship. But the answer then is the minimum number is 3. It's much harder to answer what is the optimal number of these elements that you may include. So there's no good answer here, but good news is that our brain intuitively, when trying to portray the systems or trying to portray process, actually ends up describing something in what is manageable to our brain, right? So it turns out that between 4 to 8 elements is something that's very manageable. And this toast link here actually is a TED talk that Dan pointed out to me yesterday that actually describes a little bit about this intuitive process. When you think about the systems and you try to describe the process within the system, where do you end up? It turns out we all end up in a similar place. So that's good news. So follow your intuition a little bit as you're making these decisions. As a matter of fact, don't follow your biological mode of action mind that is going to push you, to say, I'm going to identify this and this and this and this and this. So don't overburden yourself with the detail. Remember, the main objective, as Dan pointed out before, is to provide a simplified version of the biology that we can then use for the risk assessment. One thing you should aspire to, and I say aspire to because if you look at the existing AOPs, not every one of them actually does this, and sometimes it's not necessary, but you should aspire to actually including key events at each level of biological organization. And I should also point out that key events shouldn't cross between the 2 levels of organization. That's what key event relationships are for. So try to have key events structured in such a way where you're crossing different levels of organization, biological organization. Then question is, how many molecular initiating events should you have? Well, you should have as many as it requires, right, biologically. And typically it's only 1, that's the good news. But there are some instances in which you might actually have to have 2 molecular initiating events occurring before you can proceed to the next key event. And some of the examples of that come from carcinogenesis, where you might need to have mutagenicity, and then you might need to have promotion independently, and then you can only proceed to the next key event and so on. But most of the times, you're really going to deal with 1 molecular initiating event. And here's the rule to apply, right, as you're thinking about this. Both of these key events must occur. It's not one or the other. Both must occur to satisfy the criteria of being categorized as multiple molecular initiating events. The next question then is how many adverse outcomes as you're building the pathway? And the answer to that really is you could have many adverse outcomes, but it's important that these outcomes actually represent a single progression of injury, as Dan points out here. What that means is, if you're progressing from steatosis to fibrosis to cirrhosis, that's great. They can all be listed in there. But you also don't want to overwhelm the system with adverse outcomes that are not going to be terribly relevant to assessors. So it's important, as you're designating those, to include adverse outcomes that actually bear some demographic significance, right? So I can go to this example a little bit, but I think it's pretty obvious that you start with liver receptor activation, and then you progress to steatosis, which is the condition of fatty liver development. And this steatosis, through the series of steps, leads to cirrhosis, which now you're really having dysfunctional liver here, right? And then in the end, you could actually end with a hepatocellular carcinoma. So that's the kind of multiple adverse outcome situation that you're saying is appropriate. So going back to the number of the key events, and like I said, this is something that most of the biologists actually struggle with, is what is the maximum number of key events you should have? And again, there's no correct answer to this question, but remember, again, this is a pragmatic simplification. So AOP is not there to outline every biological event. It's there to simplify those. So the events you should include should ideally be events that are measurable, right? We're being very pragmatic here. Don't include something that's not measurable unless it's absolutely necessary for you to demonstrate that it's biologically plausible or something like that. So, and then also focus on taking the events that are necessary for you to establish biological plausibility, right? And I'll talk a little bit more about biological plausibility later, and so will Dan, so that will kind of become more apparent. But basically, don't use too many events. Designate the things that are measurable and things that are important, that describe the biological process well, for which there is some evidence in the literature. This is how this process works. Another thing that actually happens naturally, and you almost have to resist it as well as you develop these, is that your brain, you know, it thinks about networks and systems intuitively. So sometimes you're tempted to integrate this network into a single AOP, you know, and kind of compound things, integrate multiple key events into one key event so that you can portray multiple biological pathways and so on. Don't do that. What you should do is you should remember that AOPs are actually modular. And the reason they're modular and conceived like this is so that we can then use a network of these single AOPs that we have developed to convey the biological complexity, basically. So again, remember, AOPs are modular, you know, make them simple. But as you're doing that, it's actually a good idea to think about what other things might you connect them to and what other things may already exist in the knowledge base, because then you can actually, you can kind of leverage what you're doing of something that's already in the system and kind of help build on this massive set of networks. Having said that, it can be overwhelming, you know, to understand what is all right now in AOP-KB. So, you know, if you're overwhelmed, then stick with thinking single AOP, here are my key events, clarify it for yourself, and then maybe go into the AOP-KB, look at the things, and Steve will show you how you can do that. And then maybe adapt your AOP to fit what is in the system already. And this is where it gets really tricky when it gets to the naming of the things. So, you know, you name something and then you look at that name and very quickly decide, well, I really don't like this. How specific should the name of the key event be? And it definitely should be more specific than changes in gene expression, for example, right? That's not good enough because you're not conveying what is being measured, what is actually happening. So better is, for example, 3-beta HSD mRNA transcript abundance is increased, right? So that's better description. Another thing that kind of sometimes you want to do because you know so much biology, you kind of want to compound multiple key events into one. So sometimes the descriptions of key events, and I'm guilty of it, you know, you kind of have to curate yourself not to do this. So here's a great example here that says increased transcription and translation leading to increased enzyme activity, right? This is your knowledge of biology wanting to come out. But really, you should define this as separate key events. What you're doing is you're compounding multiple key events. And typically, this is not a good thing because it is going to make it much more difficult to share among different AOPs because others are not operating on this compounding principle. So separate your key events if they're necessary. Another very important, and this is a really critical rule when you're designing AOPs, is that the key events must be essential. And really what that means is if you have your string of the AOP, if you take one of these key events out, essential means that you cannot progress to the adverse outcome. And sometimes what happens is we have event that is not essential but occurs very commonly in response to certain stressors. So a great example of this is vitellogenin, for example. So vitellogenin is an egg yolk protein that is normally initiated upon activation of the estrogen receptor in female fish. And in female fish, it's used then for egg yolk development and so on. If you expose a male fish to something that is going to agonize estrogen receptor, you make vitellogenin, and it's a great marker of exposure to the estrogen. So if you were developing AOP, you may be tempted to include that vitellogenin induction in the AOP and say, and then this leads to male infertility, let's say. The reality is there is no empirical evidence that the increase in the vitellogenin itself actually, is what progresses to the infertility. It's just kind of a marker of some other event. So what you can do with knowledge like that, if it's really good, is you can then designate that as an indirect measure of the key event, which is ER agonism in this particular case. So just always kind of, test that essentiality rule as you're doing this. If I remove this, is it going to progress to the adverse outcome or not? And then, we told you, in a sense, to be somewhat specific. And now I'm going to tell you not to be too specific. So there's a lot of judgment going on how specific you really ought to be. But this is what starts happening as you develop these things. Well, should I name it ER binding or ER antagonism? How far should I go with qualifying the type of interaction? Should I say, it's this isoform versus that isoform? Should I say it's this cell type versus that cell type, and so on? And when deciding on the naming of the key events, you should really apply one very simple principle to make the decision, which is easier said than done, okay? But the idea here is that you're going to use the rule of functional equivalence. What this means is, if function is the same regardless of the cell type or regardless of the species and so on, do not use that in your key event specification and naming. So here's some great examples here. So for example, ER functions differently depending on whether it binds agonist or antagonist, right? So it functions differently depending on whether it binds agonist or antagonist. Well, in this case, you would specify this in the key event name. If you have an enzyme, like an example 2, that has the same catalytic activity, even though they're two different isoforms, there's really no need to specify this in the key event. The good news is that there are ways to specify some of these things, and that's what key event relationships can do for you. So for example, when function is actually equivalent regardless of the context variables, but outcome is dependent. So function is the same, right? But the outcome is going to be modulated by these variables. Then you can use the key event relationships to start describing that. So you can say, MIE in developing organism will lead to key E1, whereas in the adult organism, it will lead to key E2. So this is the way to kind of start clarifying things. Okay, so I'm going to leave it that, and we'll get you the case studies, and you can start working on those. So remember, you're supposed to think about the text that we have given you, identify the key events, and try to identify the AOP. And it's kind of unavoidable. You're going to think about key ARs as well, but you don't need to define those yet.
Okay, so I have the next 45 minutes or so to give you an introduction to the knowledge base, or an introduction and a tour through the knowledge base. So the first thing I need to do is convince you that it's necessary that we have one, because that to me is not an obvious conclusion. Assuming I'm able to do that, then I'll talk to you about the components of the existing knowledge base, and then I'm going to transition from there into a tour of the system, including actually pulling up the knowledge base and looking at it. So I'm going to start from, is it absolutely necessary to have a knowledge base to describe an AOP? And I will say no, it is not. And in fact, the first AOPs that were developed predate the knowledge base, and they were written out in documents. And those documents were systematically organized. And in fact, we've got mechanisms in place now where we define, you should put this information in this section. And it's a very quick review of the document that would be required. for to determine whether someone followed that organization. So there's no need for a knowledge base for that. Transparent and well documented. Again, that's pretty straightforward. If you put the necessary information in the necessary places and you provide the evidence you used in organizing it, that's fine. The documentation, typically these things go through rounds of review, so as long as you keep track of the versions of the document at the different stages of review and who participated in those reviews and who the authors were on the different documents, you've met the transparency requirement, and there are mechanisms in place to do all of that that do not require a knowledge base. Scientifically defensible and credible. Knowledge base will do nothing for that. That is entirely on the authors. So at this point, and in fact, the OECD Guidance, which I'll show next, really lays out the description of the AOP in the form of a document. So if you read through the OECD handbook, this is how it is going to describe that AOP. And so at this point, I'm probably not doing a great job at convincing you that there is a need for a knowledge base. However, I'll start by saying that these AOPs, when you actually print them out, are ranging between, say, 30 and 80 pages. So I'm guessing a lot of you probably have written something at some point that may have been that long, but you probably don't make it a regular occurrence. The second thing I'll bring up is in Dan's presentation this morning, you saw where he had the 4 AOPs that overlapped in the last 4 key events, right? So Dan could, and actually Dan did, because he's one of the one that predates the knowledge base. He had documents that described that, and I wasn't there, but I'm guessing copy and paste factored into that a little bit, and which would make sense. So he reused his key events the old-fashioned way, right? So now I'm imagining, he's got his 4 AOPs, he's ready to send them off to the OECD for this big review, and the paper comes out that definitively establishes the relationship between this key event and that key event in that overlap area, right? So now but they're due tomorrow, so we're gonna fix it here, right? But now we got to copy it over to all of this, and in all of that, he accidentally deletes one of his key events, or all the evidence for one of his key events in his document, right? So while yes, it's possible to do this by looking at documents, it's probably not the best practice to do this by looking at documents. And in fact, there's quite a bit of other benefits as well. One is if we put this information in a central resource that others have access to, we facilitate collaboration and crowdsourcing. So there may be experts in the world out there on parts of these AOPs that can contribute scientifically to those parts of the AOP, but those people may not care what an AOP is. They may not work in anything that deals with chemical regulation or anything that would require this level of documentation. And so by putting this thing out there, we potentially can draw in those people and get more information on these AOPs than someone who's trying to develop the whole thing. We also avoid duplicative effort. So let's say somebody else out there is wanting to do the same AOP that you are. Well, if your AOP is out there where they can say, oh, this group's already working on it, then hopefully what they would do is reach out to you and say, hey, can we help? And now both of you can contribute to one AOP, hopefully do half the work and get more benefit because each of you are going to bring a slightly different perspective to that problem. You also heard earlier today about this idea of the AOP networks, right? So we're defining them in as simple a manner as we can, but we really don't want that simplicity to actually come back to haunt us in the long run. So if we have a knowledge base that helps facilitate this integration of AOPs that maybe weren't all four developed by the same group, but yet share some of these key events, that's going to help. Okay, so integration analysis. So I'm also going to talk, as we go through the tour, a little bit about how we're trying to structure some of this information for the purpose of allowing computers to kind of mine through some of this data and give us some feedback. Accessible and searchable. So this comes back to our documents. So you might be up for reading Dan's 50 to 80 page documents, especially once you figured out that half of it is going to be fairly similar. But once we get the first 50 of these things in the AOP Wiki, you're probably not going to want to read through all 50 to figure out which one's relevant for your work. I can assure you regulators are not going to want to do that. So kind of the last piece that really makes the case for a knowledge base is we want to get it to the point where the regulator can come in and say, this is my problem, and it can come back with, well, these are the things that might help you solve it. And so now they only have to go through those things that are potentially relevant for their problem. We can now circle back and say, if we look at some of the principles that Dan mentioned earlier and how that relates to both the need for and the design of the knowledge base. So as I mentioned before, the KEs and KERs are shared. We don't want to rewrite these same descriptions, so if we make that explicit, right, and he gets that late-breaking paper right at the last minute, he fixes it one place, it magically hits all the different AOPs, his and others that use those key events. Living documents, right? So these things are going to evolve over time. I mentioned the idea of there might be experts out there that can inform your key events and key event relationships that wouldn't build the AOP and aren't going to be part of your team. Realistically, people that develop the AOP probably are not going to continue to spend the rest of their life updating these things with every new development in the process. So it may be that these things get adopted over time by people who have renewed interest in that area. And again, they don't have to have interest in the whole AOP, just the parts that are changing. And the networks. Again, one of the key components to the knowledge base is we're really trying to connect these things. So we have made this pragmatic simplification to make it easier for people to develop these AOPs, but we absolutely positively do not want that to limit our use of these things down the road. So there's actually multiple components to the AOP knowledge base. Today I'm going to focus mainly on one of those, and that is the AOP Wiki component. And the reason for that is that the AOP Wiki component has been designed specifically around the process that we're describing today in this course. But I do at least want to touch on two of the other components which facilitate, if not directly implicate, the things that we're talking about. So the AOP Explorer is the first one. And really, when we start thinking in terms of this network idea, the AOP Explorer is designed around networks. The AOP Wiki is designed around AOPs. It's designed around AOPs because it's intended to facilitate this workflow. Now, we've designed it in such a way that you can extract the networks out, but whenever you go into the AOP Wiki, you're going to be looking at one AOP at the time. The AOP Explorer, you're going to be able to go in and search that, and it's going to say how many connections do you want to pull off of this AOP. And it's going to be a way that you can go and browse through the entire network and define, okay, this is actually the space that's relevant for my program. Another thing it's going to do is we're going to be feeding hypothetical AOPs into that system. And so when you actually go to develop AOPs, it's going to be a way for you to kind of browse through the biological space and start thinking about, okay, which of these things would be AOPs that I might want to develop. Effectopedia is kind of on the other end of the system. So it structures, so the AOP Wiki, as you're going to see, structures quite a bit of information. but it leaves quite a bit of information in free text form because that's easier for most people to work with. Effectopedia is going to structure things a bit more, and in particular, it focuses on capturing these quantitative relationships. So when we start moving into consideration of dose response, and when we start getting into explicitly thinking about what data, whether it's assay data, biomarker data, help inform this AOP. That's really what Effectopedia is being designed for. There's going to be a hub that will connect these 3 modules and others. And right now the plan is to have the 3 modules and the hub live hopefully by February of next year. So going back to the AOP Wiki, so if you were to look at this and crosswalk with the OECD handbook that I showed earlier, you would see that actually all the components are here. We have over on the left-hand side our AOP page with all of our sections that are still numbered based on the handbook. And really the only distinction we have here between the layout of the handbook and the layout of the wiki is we've broken out our key events, which can be of our 3 types, and we've broken out our key event relationships. And that's the essential piece that involves reuse. But we have those things linked into our AOP. So what this allows us to do, and actually what we have done for these different review cycles, which I'll show you towards the end, is we can generate a document that actually looks pretty much just like the document you would have created based on the handbook. But by doing it this way, we now enable the reusability of these different components. So just to give you a quick tour through the knowledge, or through the wiki component of the knowledge base, I should say. So this is just going through that in a little bit more detail. Or I forgot, I think that was the end of a section. So we're going to briefly go through what the different sections are, what gets captured there, and then I'll show you what it looks like inside the wiki. So our title, the current guidance for the title, if you have an MIE and an adverse outcome, which is encouraged, you start with the MIE leading to the adverse outcome. If you need more specificity to make that a unique AOP, you can add that, but that's kind of the minimum amount. If you do not know the MIE yet, which is possible, then the guidance would be you pick the earliest key event in the process. Authors. This is actually quite important. So I mentioned the case earlier. In that case, they had the authors listed. They just didn't get mentioned. But we do have quite a few AOPs in the wiki that don't have authors listed. And so, you don't, in that case, not only do you not know who to cite if you want to use the information, and most of these early stage key events actually say don't cite or distribute, but people cite them anyway. But not only do you not know who to cite if you're going to use it, but you also don't know who to contact if you want to help, because the current best practices for the wiki is you don't start editing other AOPs unless you've gotten the author's permission. Now, key events and key event relationships are a bit more loose because they're used by other AOPs, so the guidance there is if you are going to build an AOP that uses that key event or key event relationship, that gives you certain edit privileges on those key events and key event relationships. And if there's conflict, we'll figure out whether the benefit in having a merged key event outweighs the conflicts between the different parties that are trying to describe that key event. But authors are important. So if you develop AOPs, you really do want to put in the authors. Oh, the other part about authors is, the people that enter the information into the wiki are tracked through the system by their username. But typically, you've got a lot of authors that contribute that actually aren't actually putting the information into the wiki. And so this is an opportunity for those people to get credit as well. The status, right now, you can see the under development, do not distribute or cite. So that goes up automatically when a new AOP gets created. And the reason for that is that authors were worried about this whole idea of putting my work out for the world. And one of the earliest concerns was not getting scooped, which is now coming up. But the earliest concerns were, if I'm not done, right, and somebody goes and uses this thing, I don't want to be responsible for them using something that I'm still in the process of working on. So that was why we added that. In theory, you want to remove that, the author would, when they reach a point where they feel like it's okay to use. The other thing we put in there is the OECD project that it points to. And that's just helpful in showing which of these AOPs are tied to which projects. Eventually we'll have more information here too on the review, the stage of review that these things are. So the next step is an abstract. So the idea here is to give a one-paragraph summary, just like you would for a journal article, that would allow a person to quickly review the AOP and decide whether or not it's valuable for their use. You can provide some optional background. Again, the idea here would be to keep it short. And if you have a lot of background, to link to that in other places. And if anybody's interested in how that would be done, we have an example in the wiki currently I can show you later. The next stage of the wiki page provides a short summary of the of the AOP. So you've got your molecular initiating event, your list of key events, and your adverse outcome. And you'll see that these things have the ability to show what calls have been made. So again, this coupled with the abstract is really intended to give a person a quick glimpse at, is this likely to be relevant for my purposes or not, before they start digging into all of the details. The details then go on the key event and key event relationship pages. So the key event pages will have a short description of the biology, as I went through in my breakout group. The key event itself should be a more generic description of the biology, but then it should have measurements associated with it, right? So the example would be a saturation of DNA repair versus the measurement of this particular enzyme. And so you would describe both of those processes. You would describe the biological processes. You would also sort of describe the measurements that can be used to detect those processes. You can capture the species, and eventually we'll probably add the other domains as well. Right now they're only captured on the AOP page. The two specialized key events have additional components. So the MIE, since that is the point of interaction with chemicals, have the ability to specify chemicals that interact with that MIE and evidence for that interaction. The adverse outcomes, since they are intended to be of regulatory relevance, have an optional section. You don't have to fill it out, but there is a section there for you to describe what that regulatory relevance is, and that could be previous applications. This just shows an example of the key event pages, and we'll see most of this stuff, I think, when we go through the demo. This shows the chemicals on the key event pages, and one thing that I wanted to highlight as we went through this particular section is you will notice that on the key event page itself, right, we capture chemical initiators, but that's it. So, and the evidence for them. But we don't capture any of the chemical-specific information. So again, chemicals are associated, but we don't incorporate any of the chemical-specific information on the key event page itself. And this just shows actually that most of this information, again, is captured in text form. So we have sections within the page. You edit those sections and add your information. So very similar to what you would do if you were documenting it in a word processing program. Yes, so the key event relationship pages are the same. And the thing that you should have noticed over the course of all three page types, right, is that we really break things up into these two sections, right? So we've got structured information, which captures things like the relationship between a key event and a relationship that includes that key event, and all of these AOP components and the AOPs that contain them, and the type of relationship, and the weights. So we capture some of this information in this structured form. All of this information actually can be read by computers and can be used for making inferences, whereas the textual descriptions here are really intended for human use. So what we've tried to do is strike a balance between capturing enough of the structured information that we can make this useful to people, but not making you split out all of these things into individual things and making it impossible for the person who's developing the AOP to follow it. And so then we have so once you have so now we're flipping back to the AOP page, and so you enter a table where you put in all of your connections, and then what we will do once you've done that part is we have a little program that will give you we call it a network view. Eventually it's going to have the attached AOPs, and eventually meaning hopefully this month. It will show you the attached AOPs so you can browse through the wiki. Again, it will never be a full network, but it's kind of a bridge into the AOP Explorer module. But the nice thing about the network view that we have is it really shows you, based on what you put up there, whether your AOP is matching with the guidance or not. So for example, here we've got our MIE that leads to our adverse outcome. That's in pretty good shape. Here we've got everything, but we got something over here that we're not quite sure how it fits. Here we don't even know where that's going. So here's a case where maybe this should be actually split out into multiple AOPs. So this might be a case where we've got our network now, but we really need to split that out so we can better define the individual AOPs. Okay, and then this just brings it all back together. There's an assessment down here at the bottom, which is where you're going to, once you have all of that information assembled, then you can go back and kind of review that information and provide a short, concise summary of, this is my overall impression of the AOP. This is where the data gaps are. This is where you might have issues. So again, it's kind of a short summary that captures a lot of that information. And again, makes that AOP page kind of your first stop in figuring out, is this something I want to use? All right, so the first thing we need to do is get there. And obviously an easy way is if you type into Google either AOP-KB or AOP Wiki, then it's going to come up. Or if you want to go straight there's AOP-KB.org, which will get you to this picture, and you're one click away from the wiki. There's AOPWiki.org, which would take you straight into the wiki main page. Once you are there, here, then you can read all about our effort, or if you're wanting to get straight to the AOPs, we have this link over here that provides you with an AOP list. This is where I can talk about the current OECD progress. So there's 17 AOPs that are listed in this first section, and they're technically ready for commenting. That means the authors have agreed to take off that, do not mess with this AOP yet, and open it up for commenting. The bulk of these are actually at some stage of OECD review, and I'll show you at the end how you can figure out what stage they're in. But all of these things have undergone at least one round of OECD review. There are 3 that are mature AOPs. They're pretty close to the same quality as the ones above, but for whatever reason, they have not been submitted to the OECD yet. Most of them just have certain things that are missing that the authors want to finish up before they put them in for review. So if you're looking for AOPs just to kind of browse around and see, these 17 are going to be the ones that are guaranteed to have a lot more information associated with them. When you get down into the AOPs under development, there are certainly some in this section that have quite a bit of information, but there's a lot that don't. So it's just going to be hit or miss, and how many of these do you want to click through before you find one that's a good example. Now, if you're looking for a specific question, then you can actually you can go up through the search box, as we'll show in a little bit later, and potentially find things that are under development that would be informative for your question. And so I think he started with the ovulation one. Yes. So what I've done now is I've clicked on a link to take me to a specific AOP page. And what you can see, so one thing to point out is that I have not logged in. So everything that I'm seeing now, you or anyone else in the world can see as an anonymous user. The only thing you can't do as an anonymous user is make any changes to anything. So it's view only. If you wanted to make comments on any of these things, you can self-create an account. It's pretty straightforward. It asks you some simple questions. All of them are very short answers, and if you go through the main page of the wiki, you can find the answers if you don't already know them. The only reason for that is to keep computers from creating accounts and spamming, which we had for the first month that the wiki was live. So that's very easy, and then you're able to comment on AOPs. You're not able to edit them. If you want edit privileges, you need to email, and right now you really need to be a member of an OECD project. We're hoping to open that up soon a little more broadly, but yeah, there's a slightly higher bar to be able to create or edit AOPs. So this shows you the AOP. You've got the authors up at the top, and then we've got this summary section that gives you a brief overview. This one actually has some calls for the different things, and if we get down to the network view, we can see how those calls, if I organize this a little bit better, how they're depicted in the network view. So you can see here one of them does not have a call, so it's got a very skinny connection. The two at the end have strong calls, they're very thick, and so we have a gradient along there. And so you can look at the network view and kind of get a quick overall view of what the AOP is. You can then drill down into individual key events. So we'll start with the MIE. So what we can see first is that this MIE is actually part of several AOPs. And so you could click on any one of these and see what the information specific for that AOP. We have several chemical initiators that have been tied to this key event. And because of that, we have information down here in our evidence section for the different chemical initiators, as well as measurement for this thing and a description of the biological basis for this key event. If we look at one of the chemicals, we can see some information about the chemical. So what you will see is that the chemical initiator has a table for chemicals and metabolites. There's two purposes for this. One is if you've got a chemical, especially chemicals that are metabolically activated, you can put in the chemical and its metabolites. So that's still talking about a specific chemical, but it gives you some of that information. The other reason is you might have a class of chemicals like phthalates that do certain things, and you don't want to have to put in every one of those chemicals every time you use them. So you could create a chemical initiator called phthalates, put all of your phthalates on there, and then any MIE for which all of those chemicals are relevant, you could then just tie that one initiator to it. And the other thing that this page currently captures, again, is the information about exposure. Now here I'm talking about exposure to the level of tissue dosimetry, right? Because MIEs are by definition going to happen inside the body. So when we're talking about characterization of exposure, that includes the ADME, absorption, distribution, metabolism, excretion, that affects where it goes and how much gets there. And like I said, this is the part of the knowledge base that we're really looking to revamp and think about better ways that we can capture that information. Right now, they're just here. And so now if we go back to our molecular initiating event, which is also part of, I believe, this one. And so hopefully I'm still following the slides that were in your handout. And this just shows a difference. So again, by even with the capabilities we have now, you can see how you could start one place, browse to another place, and find other related AOPs. So it's not as nice as it will be once we have the network view where it's got all of the attached AOPs there for you. But even now, there's some indication of the overlap among these things. And so here again, we see there's a different set of key events, but we do have some that are in common. If we take a look at an adverse outcome, just to see what those look like, we can see that, in fact, this adverse outcome is the case for most of the cyclooxygenase inhibition MIEs. So now if we think in terms of networks, right, what are we expecting here? We've got cyclooxygenase inhibition, we've got reproductive success reduced as our adverse outcome, right? And we're going to have a pretty nice network of things. So, and because these things are defined the way they are, that network will self-assemble. So even though it's not necessarily obvious when you're looking at each individual AOP, it is captured inside the knowledge base. And like I said, we will get better at displaying that information. The Explorer will do a great job at displaying that information. And so over time, it's going to become more and more valuable. We have a few other structured things on this page that you can fill out if you are so inclined. And then we have the individual sections. And again, because this is indicated as an adverse outcome in at least one AOP, it has this added section where you can describe the regulatory examples. And now I'm trying to remember where. Oh, yes, so now we were going to go back and show the search. So if we come back here and look at estrogen receptor antagonism as an example, so for one thing, I can show you that this is a more advanced AOP than the other ones, and so it's got quite a bit more information down here. Oh, you also have the ability to put in a self-generated diagram. If you want to use this particular template, we even provide a PowerPoint version that you can download and edit to match your AOP, and then re-upload. It's easiest if you do a JPEG file. And then you can see the overall assessment that went along with this. And then the last step was to search estrogen. Oops. So if instead I want to come in and say what all is in this knowledge, or at least this wiki portion of the knowledge base that relates to estrogen, I can do that. I'm going to get pages back. Now, in this case, this is an older one, so it has a lot of title matches. To be honest, most of the stuff that's going to come back looks more like this, where we've converted the names to be unique identifiers. But even here, you can get the information that you want. You know whether it's an event. You know whether it's an AOP. I'm just going to come back up here because we wanted to show the relationship page here that relates to this particular example. So again, we've got the upstream event documented, the downstream event. We can follow these links to get to those pages, and then we have the AOPs that include that information. And then the last thing I want to show you, which is not in your handout, is just the OECD review process. And for that, I'm going to grab this one because I know it has been through both stages. So again, for all of those AOPs that are in that top set of 14, they've all undergone at least one stage of review. I didn't talk about commenting earlier. Maybe I'll save that for the end of the day or later if people are interested. But all pages have a discussion page. And if there's nothing on the discussion page, you just add comments. Feel free, once you've got that self-created account, feel free to add comments to any page in the wiki. The AOPs that have undergone formal review have this tabbed structure. So if you want to add comments, which I wouldn't be able to do because I'm not logged in, but I would click here. There's instructions on the main page showing you how, but there'd be a spot. Maybe I'll show that this afternoon when I am logged in. But you can see all of the formal reviews without being logged in that have taken place. So in this case, it was reviewed internally by the OECD Extended Advisory Group, and it's in the process of an external peer review. These internal and external reviews, there is a formal process for both the review itself and for responding. So the review itself is actually stored in the wiki. So you see the comments both from the reviewers, and somewhere down here we had one that the author responded to. Here's one where the reviewers had more substantial comments, and the author has a response that they have put in addition to the edits that they made to the document. They have provided an official response to that review. As I mentioned, these reviews, right, as you might imagine, you can't review something that's constantly changing. So what we've done is we've captured snapshots. The next iteration of the knowledge base will make that a little easier so that everybody can create snapshots. But for this, we created them separately. So we have PDFs that correspond both to the thing that was reviewed by the 3 people listed here, as well as the revised document that was reviewed by the full external advisory group in May. Actually, it was reviewed in June. It was created in May of last year. And based on that, a subset of them were selected for external review. This was a combination of both the external advisory group making the recommendation and the author being comfortable that they either could make the needed changes or that they were comfortable with it being externally reviewed. This particular AOP was, so we have a separate page that captures that external review. In the case of the external review, the reviewers were anonymous, so there is no reviewer information, but we do have the reviewer's comments as the reviewer submitted them, and we do have responses from the authors, and as you might imagine, this was a separate snapshot. So we now have 3 snapshots that have PDFs. Within that PDF actually are links to the static version of the wiki pages, so you can, in mechanisms that are beyond the scope of this, you can go in and see what the differences are between what was reviewed and what exists now.